We're going to do a mini unit on bacteria and viruses. I'm just going to give you some background here, and then we will do the bacteria lab and go forward after that. Remember that bacteria, we just finished talking about all this, there are two main kingdoms. We have kingdom Eubacteria, and those are the ones that have the cell wall made of peptidoglycan, and then we have domain Archaebacteria, kingdom Archaebacteria, and those are the ones that we called extremophiles. They live in really hot, really cold, really salty, or environments with no oxygen. General bacteria structure looks a little bit of something like this. Remember that bacteria are prokaryotes, so they have no nucleus. They do have an area where the chromosomes are housed, just kind of hanging out in the middle of the cell, called the nucleoid. Bacterial cells are surrounded by a slimy layer called a capsule that protects it from getting broken down. And then there are little hair-like structures called pili that allow it to exchange genetic information with other bacterial cells and sometimes help it move things across the cell surface. Bacteria are very small. Prokaryotes, as a rule, are much smaller than eukaryotes, so they are much smaller than a human cell. There are three main ways that we identify bacteria. First, by the shape, then by the cell wall type, and whether or not they move. So what you see here are the three main shapes. The spherical shaped are called the cocci, the rod shaped are bacilli, and the spiral shaped are called spirochetes. Bacteria reproduction, it's basically mitosis. The process is called binary fission, and that's what you see right here. The DNA gets copied and the cell pinches inward on itself in the middle and divides. The other thing that bacteria can do, and this increases variety in bacterial cells, is this process called conjugation. I don't know how well you can see it right here. That's not going to help. I'm going to circle it myself. Right here, there's a little red line in between these two bacterial cells. This is a pili, one of those little hair-like structures that I said allow communication between the cells, or exchange of genetic information between the cells. That is conjugation. Let's talk about bacterial infections. Bacteria are commonly thought of as being the bad guys, and that's the case a lot of the times. You can see right here in this data table lots of bacterial diseases that are caused by bacteria. So we have STDs, which are now called STIs, syphilis, gonorrhea, chlamydia, respiratory diseases, strep throat, pneumonia, tuberculosis, anthrax, bronchitis, skin diseases like acne, infections of wounds or burns, digestive tract diseases, any kinds of food poisoning, nervous system diseases, botulism, tetanus, meningitis, and then other diseases like Lyme disease and typhoid fever. These are all caused by bad bacteria, but there are good bacteria also that are really important for our ecosystems and for our lives. So, first of all, if it weren't for bacteria, plants couldn't use nitrogen. They need nitrogen in order to live, and there's a ton of nitrogen in the atmosphere, but the plants can't use it in gas form. So there's bacteria underground in the soil that takes the atmospheric nitrogen and converts it for plants into a form that they can use. For animals, bacteria in animals is good. We talk about different genetic modification with animals. Bacteria is good for us and for other animals for breaking down food and processing food in our digestive systems. For foods, we talked about fermentation way back in first semester, and there are lots of foods that are produced by bacteria. Those fermented dairy products such as sour cream, cream cheese, cheese itself, yogurt. And then finally, bacteria can be used in medicines. We talked about genetic engineering, how bacteria can be used to produce human insulin to help people with diabetes. Now, the true bad guys are the viruses. Those are the true bad guys. There's never a good virus. We even tried it with gene therapy and it didn't work. Viruses are, if bacteria are tiny, viruses are teeny, teeny, tiny. They're much, much, much smaller than bacterial cells, which are much smaller than eukaryotic cells. So their origin, where we think they came from, parts of cells. They're not truly alive because they're not cells. Here's some examples of viral diseases. So other sexually transmitted diseases like HIV, genital herpes, childhood diseases, measles, mumps, chickenpox, respiratory diseases like the cold and the flu, skin diseases, warts and shingles are viruses, digestive tract diseases, gastroenteritis, so food poisoning kinds of symptoms can be caused by viruses as well, nervous system diseases, polio, viral meningitis, rabies, and then other diseases such as smallpox and hepatitis. So these are all virus-borne diseases as opposed to the bacterial diseases. Those are confused quite a bit. Virus structure, there are many varieties. They're kind of funky looking. This guy up here kind of looks like a satellite, what you would see. And this one as well, maybe an orange with little spikies coming out of it. If you used to watch Jimmy Neutron, there was an episode about viruses, and the virus looked like this. The only problem with that episode is that this kind of a virus infects bacteria, and I think it was going after Jimmy Neutron and his friends. So that's the only problem with that, but that is in fact what a virus looks like. It looks like a robot. And then there's this cylindrical guy as well. Not as exciting as the robot guy for sure. Two different cycles for viruses to infect. Don't worry about these too much. The lytic cycle is where the virus injects itself into its DNA, into the host cell. That's what you see here. The virus attaches, it injects its DNA in. It basically uses that host cell to produce more copies of virus, and then those viruses are assembled, and the cell then bursts and releases all those new virus particles into the organism. The lysogenic cycle takes a longer time. So this lytic cycle is what happens like with a common cold. The lysogenic cycle is when you have the same thing happens to start here, that you have the virus attaching itself to the outside of the cell. It injects its DNA in, but in this case, now we see what happens is the DNA becomes part of the host cell DNA, and that viral DNA gets copied with the host cell DNA, and then at some time down along the road, the viral DNA starts producing more copies, and then the viruses are assembled, and the host cell bursts and releases those viruses into the bloodstream of the host organism. What you see with that, like a herpes virus or chicken pox, those viruses that go latent, but under times of stress, your immune system just can't keep up, that's when you see those show up. Then we have the retroviruses. Retroviruses have RNA as their genetic material instead of DNA. The most common example that we talk about is HIV, human immunodeficiency virus. Uses RNA instead of DNA. These are crazy. These are neither bacteria nor virus, but they deserve being talked about. They're called prions. Don't know a whole lot about them. We know that they are mutated proteins, and they cause big problems in the nervous system. CJD is Creutzfeldt-Jakob disease. That's of humans. Scrapie is a prion-related disease in sheep. And BSE, bovine spongiform encephalopathy, this guy right here, You may have heard of this before. Mad cow disease. Mad cow disease. Somehow these mutated proteins convince other proteins around them to become mutated. They brainwash the other proteins and send them out and misform them and cause lots of trouble in the immune system. So that's it for bacteria and viruses. We will get started with the bacteria lab in a couple days.